Time-lapse microscopy allows the visualization of the processes of development. Growth or drift of a sample that occurs over this image acquisition can result in an inability to follow or track the cell movements. The following protocol will use open-sourced imaging processing software to correct for three-dimensional drift over time. Capturing a series of optical sections at different focal planes allows the generation of a three-dimensional model of a sample. This can be extended into 4 dimensions by creating a time-lapse series of 3D datasets. In long-term time-lapse experiments, it is common to observe sample movement. This can be caused by slight inaccuracies in the hardware controlling stage and focal positions, while in other cases, drift is a result of movements induced by sample growth or flexibility within the mounting media. Methods exist to compensate or limit these movements, with many improvements to hardware focusing systems and the increased rigidity of mounting medium. However, these approaches cannot be applied in many cases due to the imaging setup required to provide suitable conditions for the sample's maintenance and growth. To correct for sample drift, we have developed a plugin to utilize the open-source imaging processing platform VG. Our Correct 3D Drift plugin is able to perform phase correlation registration to correct for movement that occurs as a result of sample drift in three-dimensional time-lapse experiments. In multi-channel experiments, the Correct 3D Drift plugin utilizes one channel to determine the required correction. This correction is then applied to any additional channels, resulting in the registration of the 4D dataset.